So, I'm sure a lot of you guys have heard about DNA, and I'm sure a lot of you guys know what it does in the body. It's a cliche, but it's the blueprint of creating everything in our body. It has the instructions for creating everything from our bones to our muscles to our organs, down to the smaller scale, like all of our organelles and our cells. And most directly, it creates the proteins that serve as building blocks to create everything else that I just mentioned. But for creating something super big and complex, DNA is, we can't see it. It's not that it's tiny. Each cell has over 6 feet worth of DNA, which is like my height in each cell. So it's like pretty tall. Like there's a lot of it. And if it wasn't super compacted, by chromatin, histone proteins, that's a whole different story. But if it wasn't so greatly compacted, it would be a lot larger than what we normally would think of it as being. But in reality, due to it being super compacted, DNA is like in the scale of micrometers. It's like 1,000th of a millimeter, which is ridiculously tiny. It's microscopic, like we can't see it with our bare eye. So how do we measure DNA, or tell how much DNA is in a sample? Like it's too small to see. So what tool do we use to see it? So the tool of interest today is a nanodrop, and a nanodrop measures DNA concentration in a sample, meaning it measures how much DNA is in a certain volume of a sample. So it does this by shining a light through the sample, and kind of like how you would flash a light through a glass that maybe isn't fully transparent, you can tell how much of the light goes through the glass and how much of it is absorbed. That's exactly what the nanodrop does. It shines a light through the sample, and it can tell how much of that light is absorbed by the DNA in the sample. So I can explain the specifics of how nanodrop works, I need to go through a couple of main parts of the machine. So the most basic and probably one of the most important parts are the two pedestals. You have two pedestals. One of them is on the base of the machine, and one of them is on the arm that you bring down to start the measurement. So you place your sample on the bottom pedestal, and you bring down the top pedestal that's attached to an arm so that it sandwiches the sample in between the two pedestals. And the pedestals are important because there's light that's going to be shining through the pedestals, through the sample, and that's how you get your measurement. And this light comes from a xenon lamp in the machine. There's xenon in the machine, like that's the stuff in nuclear reactions, like that's actually crazy. And that's your light source. And your light source travels through fiber optic cables, which kind of serve as like the transportation, means of transportation for the machine. It goes through the fiber optic cables from the lamp down into the sample, and then what's recording, how much light's being absorbed is something called a photodetector, and it works with another machine called the spectrophotometer. And basically, all you need to know is that these two things work together to determine how much light is traveling through the sample and how much of it is being absorbed by the DNA. And it records this, and it can. These are the measurements that it uses to determine the absorbance and the concentration of the DNA. And basically, it sends these measurements as raw signals through another fiber optic cable back to the software of the machine, which does the rest of the calculations to determine your final concentration, how much DNA is in your sample. So there's a lot of really cool, crazy stuff going on in this one small machine. All right, so now I'm going to give you guys a bit more of a specific explanation of how the nanodrop collects the values and converts them to a concentration. So once the light is shining through the sample, the photodetector, the spectrophotometer, it collects the data, it collects how much light is being absorbed at 260 nanometers, which is a wavelength that DNA is known to absorb light. So it collects how much light is being absorbed at this wavelength and stores it in a value called the A260 value, which is absorbance at 260 nanometers. Once it has this measurement, it sends the data it has by an electrical signal through fiber optic cables back to the computer mainframe of the machine. And once in the software, the computer plugs this value into a formula to calculate concentration. And this formula is your A260 value divided by your extinction coefficient, which is the amount of light DNA can absorb at a certain wavelength. And this is a constant. So it kind of depends on the wavelength you're looking at. And then you divide that by the path length as well, which is the distance that light travels through your sample. So it's a pretty basic formula, but the means of acquiring the data and then getting it back to the computer to calculate is simply amazing. But in reality, it's just all plug and chug, baby. All right, so now that you guys have sat through my explanation of the Nanodrop, let's go see it in action.
So right now, I'm adding the water sample to the Nanodrop machine using a micropipette, which allows me to administer small doses of a liquid. And it's really important to be precise when adding in the drop because the pedestal to add it onto is very small. And here's a closer view of me aiming the drop onto the bottom pedestal. And you can see that the drop is a very small amount and that surface tension is crazy. So you can see the bottom pedestal that the water is resting on. Behind that pedestal, there's a black dot, which is a sensor that records how much light is absorbed by the sample. And in other words, that is the photodetector. And above on that arm is the top pedestal. And that arm can be brought down on top of the bottom pedestal to sandwich the sample. I use water as a blank to give the machine an idea of what a sample with no DNA looks like before I put in an actual sample. And blanking is always necessary. It's really important to clean the pedestal after you've added in a sample and you're done with it and you have to change it out. So you have to wipe it down with a Kim wipe. And now I'm adding in my DNA, the real stuff, and I'm going to add it in the same way that I added in my water on top of the pedestal with a micropipette. Once I put in the DNA, I can bring down the arm to run the machine. And now the Nanodrop will shine the light through the sample and give us the concentration of the DNA in the sample, as well as various absorbance values at different wavelengths that is important to calculate. So this reading shows a graph of the amount of absorbance at certain wavelengths, and you can see that the max absorbance occurs at 260 nanometers. There are some other important measurements, as you can see on the top right of the screen. The Ng per microliter shows the concentration. Those are the units it's measured in. And then they have two absorbance readings from 260 to 280 and from 260 to 230, which shows some other significant wavelengths that DNA tends to absorb light at. So these are the measurements that the Nanodrop gives you.
Guys, thanks so much for watching the video. Hope you guys enjoyed. I had a lot of fun making it. It was some cool stuff that I went over. And if you enjoy the content, please go ahead and support me. Any support will do. And I'll see you guys soon.